Hello everyone, today we're going to be studying the topic cell structure and organization from Edexcel AS level biology unit 2. Let's begin. So what is cell theory? All living things are made up of one or more cells. That means that cells are the basic units of life and also the basic functional units, and new cells are made from pre-existing cells from the process of mitosis and meiosis, which we will cover in another video. So some common features among all cells are cell membrane, cytoplasm, DNA, and ribosomes. For example, no matter the cell, either it's prokaryotic or eukaryotic or whatever the cell type is, you will always find these four features in them. So different cell types contain different structural elements and combination of organelles. Like in prokaryotic cells, we have no internal membranes. Like they would have a cell surface membrane that would surround the cell, but within the cell there would be no organelles that would have their own membrane. Like mitochondria, it has its own membrane, so that won't be present in prokaryotes. But eukaryotes have plenty membrane-bound organelles. When observed under a microscope, it's possible to see the ultrastructure of different cell types. Ultrastructure is you'll be able to see the organelles and other internal structures and stuff. Level of organization. So we have organelles, which are specialized subunits with specified functions. Different organelles make up the basic structural and functional units of all forms of life, which is a cell. Then similar cells make up a tissue. Then similar tissues make up an organ, and different organs make up an organ system. Over here I have an example of digestive system. Digestive system is made up of organs like mouth, esophagus, stomach, pancreas, liver, gallbladder, small intestine, large intestine, and anus. These organs are made up of tissues like epithelial tissue, connective tissue, muscle tissue, and nervous tissue. And these tissues are made up of cells like epithelial cells, stomach cells, intestinal cells, rectal cells, etc. And these cells are made up of the organelles like nucleus, mitochondria, RER, SER, ribosomes, Golgi, etc. Okay, so we have three main domains. All three share a common ancestor from long back. That's why we have those four common features that exist in all cells. We have archaea, bacteria, and eukarya. Archaea and bacteria are prokaryotes, meaning that they have no membrane-bound organelles, and eukaryotes are just eukaryotes. So here's a eukaryotic animal cell. We have microvilli, Golgi body, microtubules, cell surface membrane, cytoplasm, SER. We got nucleolus, chromatin, nuclear pores, and nuclear envelope all within the nucleus. We got RER, mitochondria, lysosomes, centrosome, which is just two centrioles which sit at right angles to each other. Here's a eukaryotic plant cell. We have chloroplast, Golgi body, Golgi vesicle emerging from the Golgi body. We got cytoplasm, mitochondria, envelope and grana in the chloroplast, ribosomes, microtubules, RER, and again within the nucleus we got pores, envelope, chromatin, and the nucleolus. We have SER, cell membrane as our second layer, and cell wall as our first layer. We got cell sap within the vacuole, which is surrounded by tonoplast. And finally we got plasmatous matter. Cell membrane. The cell surface membrane controls what enters and leaves the cell, which means it is partially permeable and has selective permeability, helping maintain a stable environment inside. It supports the cell's shape and helps cells recognize each other by glycoproteins and glycolipids, which are present on the membrane. Substances can pass through it by diffusion, osmosis, or active transport. You can see how it looks when we see it under the electron microscope. We got nucleus. The nucleus is the control center of the cell, storing genetic material, which is the DNA, and directing cell activities like growth, metabolism, and reproduction. It regulates which proteins the cell produces, ensuring proper function. I hope you all understood this. It's pretty straightforward. And on the side we got the picture of a nucleus under an electron microscope. The nucleolus. It's a small dark structure inside the nucleus that produces ribosomes. These ribosomes later move on to the cytoplasm or attach to the RER to help in protein synthesis. It's that dark patch that you can see in the picture. Ribosomes are tiny structures consisting of a larger and a smaller subunit responsible for making proteins by assembling amino acids in the order specified by the cell's DNA. They can float freely in the cytoplasm or attach to the RER. In the picture, they are attached to the RER. So, rough endoplasmic reticulum. It's the primary site for initial modification of proteins and their transport. Proteins are synthesized by ribosomes attached to the RER and undergo basic modifications such as folding into their correct shape, formation of disulfide bonds for structural stability, addition of simple carbohydrate chains. Once modified, proteins are packaged into vesicles and sent to the Golgi. SER. Its main function is the synthesis of lipids, steroids, and carbohydrates, which are then sent to the Golgi so that they can be attached to the proteins for complex modification, as well as the metabolism of exogenous substances, which are just substances that come from outside of the organism, such as drugs or toxins. SER helps store calcium in cells. Golgi apparatus. Final processing, complex modification, sorting, and packaging for delivery. The Golgi has specialized enzymes and compartments which are called cisternae for advanced modification, like adding complex sugars, adding phosphate groups, sulfation, or addition of lipid groups, sorting proteins into vesicles for their specific destinations. Packing and vesicle in Golgi and RER is so that the substance does not react with anything before reaching its destination. Also, please remember the shape, they ask about that often. It's flattened, curved sacs decreasing in size. Mitochondria are the powerhouse of the cell, producing energy in the form of ATP. Enzymes in the matrix carry out aerobic respiration. They are vital for powering the cell's activities. It has its own circular DNA, which is called mitochondrial DNA, which is present in the matrix. Matrix is like the cytoplasm of the mitochondria, and the organelle is rod-shaped. Foldings of the inner membrane form cristae, increasing surface area to volume ratio for the respiration process. Lysosomes. Lysosomes are small sacs filled with enzymes called lysozymes that break down waste materials like old organelles and harmful substances in the cell, keeping it clean and healthy. For example, when our phagocytes engulf a pathogen, to destroy that pathogen, the lysozymes of the phagocytes will burst to destroy the pathogen. I couldn't find a proper picture, but it kind of looks like this. Lastly, we have centrioles. Centrioles are part of the centrosome. Two centrioles, or this pair together, is a centrosome, and the centrosome is responsible for organizing microtubules which group to form spindle fibers, which pull apart sister chromatids in cell division. Microtubules produced by the centrioles are also responsible for maintaining cytoskeleton of the animal cell. Cytoskeleton is like the network of proteins and filaments that hold the structure and the shape of a cell. Centrioles are only found in animal cells, you won't see them in plant cells. So here we have a prokaryotic cell which has no organelles, which are surrounded by a membrane except for the cell's surface membrane itself. So organelles that you will always find in a prokaryotic cells are cell wall, which is made up of peptidoglycan. We have cell surface membrane, cytoplasm, circular DNA, which is the main DNA, consisting essential genes needed for growth, metabolism, and reproduction. The DNA is found in the nucleoid, since bacteria don't have a nucleus. Lastly, we got ribosomes, and organelles that you might find in some prokaryotes and not always present are flagellum, which propels the bacteria to help it move. For the bacteria that don't have flagellum, just rely on floating randomly, also known as passive motion for movement. We have capsule, which is kind of slimy and sticky. It can help in protection and if the bacteria has to stick to something. We have mesosomes. Mesosomes are infoldings of the cell surface membrane, which increases surface area to volume ratio for better photosynthesis or respiration and nitrogen fixation. We got plasmids. These are small circular DNA which are not the part of the main chromosomal DNA. The difference between chromosomal DNA and plasmid DNA is that plasmid DNA only contains extra genes that are not really necessary for the organism, such as antibiotic resistance, but chromosomal DNA carries all of the information needed for the growth of the organism, development of the organism, and reproduction. Lastly, we have pili. Pili help in conjugation, which is like sexual reproduction for bacteria. Like transfer of genetic information takes place by the help of pili. Pili also help the bacterium to stick or adhere to other surfaces. Here are some functions. Capsule protects the bacteria from drying out and the attack by the cells of the immune response from host cells. Like if bacteria is infecting a cell and the host cell does something to prevent the bacteria, like attack it, so the capsule will help the bacteria. Cell wall made of peptidoglycan and murine and provides support and structure. Pili attaches to other cells, is also used for transfer of genetic material. Ribosomes involved in protein synthesis. Ribosomes in prokaryotes are 70s, which means they are smaller than eukaryotic ribosomes, which are 80s. 70s and 80s are like relative unit for size of the ribosomes. Just know prokaryotic ribosomes are smaller than eukaryotic ribosomes. We got circular DNA, which contains the genetic information needed for the cell's growth, reproduction, and metabolism. It is present in the nucleoid. Plasmids are additional genes that provide advantages such as antibiotic resistance or the ability to metabolize unusual substances, and the plasmids are exchanged through conjugation. We have flagellum, which is a whip-like structure for movement. Also, please remember how to draw all of these. Here we have parts of a light microscope. Not that important, I guess, but it will be better to know some functions for understanding. We have eyepiece from where we look, coarse adjustment wheel for initial focusing, fine adjustment wheel is for more focusing when viewing under high magnification. We got our mirror and light source. Aperture helps focus light from the light source entirely on the specimen. We got stage where we place our specimen. We have 3 objective lenses, each of different magnification. Rotating nose to help you select which objective lens would you like to utilize. Magnification. What is magnification? Magnification is how many times bigger the image of a specimen observed is in comparison to the actual real-life size of the specimen. Magnification can come from eyepiece and the 3 objective lenses. To calculate the total magnification coming from the microscope, we use the formula total magnification is equals to magnification of eyepiece times magnification of objective lens. But if in the paper they give you a picture of some microorganism and tell you either its magnification or actual length, then you can use the formula magnification is equals to image length divided by actual length. You use a ruler for the image length. Magnification and resolution. Magnification is the ability to make small objects seem larger. Resolution is the ability to distinguish 2 objects from each other. Like if resolution is too low and there are like 2 dots next to each other, you won't see them as 2 different dots. You will see them as one blurry big dot or something, and you'll be able to see clear 2 different dots if the resolution is high. Hopefully you understand. Magnification and resolution in light microscopy. Magnification is achieved by using a combination of objective and eyepiece lenses to enlarge the image of the specimen. Resolution is limited by the wavelength of visible light, which is around 200 nanometer, meaning objects closer than this cannot be distinguished. Electron microscopy. Magnification is achieved by using electromagnet lenses to focus a beam of electrons on the specimen, producing a much larger image. Resolution is much higher, about 0.1 nanometer, because electrons have a much shorter wavelength than visible light. This allows detailed views of ultrastructure. Staining your specimen. Staining helps in identifying and studying specific structures or organelles more effectively. In light microscopy, increase contrast between different parts of the specimen, making structures more visible. highlight specific features. For example, we use methylene blue, which stains the nuclei. In electron microscopy, specimens are stained with heavy metals like lead and gold to scatter electrons and improve contrast in the image. These are some conversions which will be of great help. Let's take centimeter as base unit. To convert centimeter to kilometer, we multiply our number by 10 to the power negative 5. To meters, we multiply by 10 to the power negative 2. To millimeter, we multiply by 10 to the power 1. To micrometer, we multiply by 10 to the power 4. And to nanometer, we multiply by 10 to the power 7. To go opposite, just divide instead of multiplying, like from nanometer to centimeter, just divide by 10 to the power 7. Let's look at some questions. Organisms can be classified into one of three domains. Organisms belonging to two of these domains have prokaryotic cells. Bacteria are one of these domains. Name the other domain that has prokaryotic cells. So we have three domains: archaea, bacteria, and eukarya. They have asked for prokaryotic domain other than bacteria. So our answer is archaea. The diagram shows the outline of a bacterial cell. Draw three labeled features on this diagram that may be found in a prokaryotic cell. So the cell wall is already drawn. We can draw any three organelles beside the cell wall. You can draw any three among capsule, villi, membrane, plasmids, DNA, ribosomes, and flagellum. I drew all of them for your understanding. So the diagram shows the structure of adult hemoglobin. We have two beta polypeptide chains and two alpha polypeptide chains and a heme group. Describe the role of the rough endoplasmic reticulum in the synthesis of hemoglobin. So since polypeptides are proteins and proteins are formed from the ribosomes on RER, and then the chain of amino acids which is formed by the ribosomes will go into the RER, and then it will be folded into its shape and it will be transported out from the vesicle, which is also produced by the RER. So the photograph shows yeast cells using light microscope and an electron microscope. Which structure identifies yeast as a eukaryotic organism? So remember, prokaryotes have no membrane-bound organelles and eukaryotes have membrane-bound organelles. So we need to find a membrane-bound organelle. Let's look into each of these. So we have a nucleus, we have J. J is curved sacs, but they're not decreasing in size, and it's next to the nucleus, so it's RER. We have K, which is cell membrane, which is present in all cells. We have L. L is cytoplasm, so that is also present in all cells. And we have M. M seems cell wall, but cell wall is also present in bacteria. So our answer should be J. J tells us that it's a eukaryotic organism. Explain why structure J can be seen using the electron microscope but not the light microscope. So it says RER is too small and light microscope cannot provide enough magnification to see it. Only the electron microscope can. Explain why the nuclear envelope cannot be seen as two membranes using the electron microscope. So distinguishing 2 points was resolution. We can simply say the membranes are too close together and the resolution of this microscope is not high enough. A student investigated the mitotic index of cells in the tips of tendrils. Acetic or seen stain was added to the tissues from the tips of the tendrils. Give one reason why the cells were stained in this investigation. So stain is just to create contrast between different parts of the specimen. The photograph shows some species of a flowering plant native to South Africa. Which of the following is the domain for this plant? So we know plants are eukaryotes, so C. The table below shows some features of prokaryotic cells and eukaryotic cells. For each feature, put one cross in the appropriate box in each row to show whether the feature is found in prokaryotic cell and eukaryotic cell. Prokaryotic cells only, eukaryotic cells only, or not found in either. So if you remember, I talked about some common features between all types of cell, no matter if they are prokaryotes or eukaryotes. And cell membrane and ribosomes were among that list, so they are found in both. The Golgi apparatus is found in eukaryotic cell only. In the space below, draw and label a diagram to show the structure of Golgi apparatus. So you can draw something like this. Flattened curved sacs decreasing in size. We got cis phase and trans phase. We got cisternae, lumen, and secretory vesicle. In a multicellular organisms, cells can be organized into tissues, tissues into organs, and organs into organ systems. Give one difference between the structure of a tissue and that of an organ. This is pretty simple. If you remember, tissues are made up of similar cells and organs are made up of multiple tissues. Alright, thank you. Bye bye.